I will share the first insight from our case study on blue carbon potential of shellfish reefs. This is a joint work which was carried out with my colleague from AVI, Dr. Bernadette Pogoda, and with Bill, who is also present here from Harriet University. And this work has been done under Define 2 project, which is funded by BFN. And today we are going to talk about the organic and inorganic carbon seasonally and with increasing sediment depth in a shellfish ecosystem. When I say organic carbon, it means TOC or total organic carbon. And when I say inorganic carbon, it means PIC or particulate inorganic carbon or carbonate. And we will also discuss what this organic and inorganic carbon fraction deposition it means for restoration management and realistic blue carbon accounting. So before I start, our earlier talks it already covered the definition of shellfish reefs and what is blue carbon. So I'll go straight into the debate, that is blue carbon, the shellfish reefs, do they act as carbon source or carbon sink? Now, to understand and to answer this question, we need to look into different biogeochemical processes that happens within a reef ecosystem. So, reefs they affect carbon via opposing pathways like filtration or biodeposition. They add organic carbon to the seabed, whereas microbes they degrade or remineralize it, and calcification it builds carbonate in cells, but respiration it generates carbon dioxide. So whether these reefs they look like source or sink, it depends on community composition, the hydrodynamics of that particular location, the seasonality, and the balance between organic preservation versus organic carbon turnover. So together with carbonate production and maybe possible dissolution of cells. So based on the premises, our goal was threefold. The first goal was to quantify organic and inorganic carbon storage within these ecosystems. The second was to track how carbon change with season and with increasing sediment depth. And the third was to generate a baseline data for restoration and accounting framework. So based on these goals, we selected two objectives. The first was to estimate the carbon standing stock of reef habitats, and the second was to establish annual carbon budget of individual reef building organisms. So, this coming to the study area, we had a total of two study locations. The first one was in Zurich in Germany. So it borders the North Sea. You can see the locations which is within the rectangle shape. It is a very small island called Zurich in North Sea in Germany. So we collected the sample during low tides across 4 seasons during summer, autumn, winter and spring. And these 4 seasons were considered because we wanted to account for the blooms, the spawning, and the hydrodynamics, which strongly control organic inputs and cell growth. So this is how the intertidal reef it looks during. The picture was taken during low tide in 2024 spring, and based on physical observation, we selected 6 different habitats within this reef. So the first one was blue mussel dominated mixed bed or S1. The second one was Pacific oyster dominated mixed bed or S2. Then the third one was intertidal sediment, which is S3. This was taken as control. And the fourth one was seaweed associated mixed bed. This is a fucus species, brown algae. And the fifth one was tide pool or S5, and then reef adjacent subtidal sediment or S6. Now this subtidal site It will help us to test the export or retention of carbon and the effect of continuous submergence. Uh, yeah, and here the key contrast is between the biogenic structure, which is formed by reef or algae, versus the exposed sandy flats. So we will know where carbon accumulation happens more. So coming to our next locations, which is in Loch Ryan in Scotland. So Germany currently lacks any subtidal shellfish reefs, proper subtidal reef. So we added a natural Australis habitat, which was in Loch Ryan, and we collected sample during June 2024, and only a single time sampling was carried out by divers, and this was carried out with the help of a bill from Hyderabad University. And this let us compare species and see how subtidal conditions it affects accumulation and preservation of carbon, and how they differ from intertidal systems, both ecologically and biogeochemically. So this is our team. We were collecting samples in June 2024. And then this is a very small video where the natural habitat of Australis it is shown. I hope you can watch the video. Yeah, so this is how it looked. And then we come to the methodology part. So in the field, we isolated two layers. One is biotic layer that is formed by the organism, both their tissue and their cells. The tissue it stores small amount of organic carbon for a brief period of time, whereas the cells they can store large amount of inorganic carbon. We also collected the underlying sediments, and these sediments they can store up to kilograms of carbon per square meter over depth and over time if the retention of carbon it exceeds the export of carbon. So we collected top 30 centimeter of sediment, and the sediment stock of carbon it was computed by integrating percent of carbon with sediment bulk density of 2 centimeter thick slices, which capture the surface pulses during the bloom, and also rapid changes in permeable sands. And the biotic carbon it was calculated from the aerial densities of organisms and percent of carbon which was stored within their tissue or within their cells. And whenever there were macroalgae present, the amount of organic carbon it was integrated during the calculations. So I will show you the another video which shows how we collected the sediment cores in subtidal ecosystem. Yeah, this is how we collected the sediment cores from the subtidal area by diving. Now, coming to the abundance of blue mussels and Pacific oyster at Zult, these four panels they indicate 4 seasons of abundance of oysters and blue mussels, Pacific oysters and blue mussels, and we see that a strong seasonality of abundance with a strong spring recruitment in case of both blue mussels and Pacific oysters. And mussel beds in general they show a higher density compared to the oysters, and but oysters they are increasingly appearing in mixed patches. And the takeaway message here is more filter feeder they have more potential for biodecomposition or more organic carbon delivery to the sediment, and also more shell production over time. Coming to the abundance at Loch Ryan, so we see that European oyster their abundance was between 75, 80 to about 175. In average, they were about 80 individual per square meter. So that was quite good. And they consisted of both juvenile and adults. About one third of the population they were juvenile. So then we come to the condition index or the health of the organisms. So in the left panel, we see the condition index of blue mussels, and in the right panel, we see the condition index of Pacific oysters. So we can see that condition index it peaked with the spring bloom in case of both blue mussel and oysters, and blue mussels they generally showed a higher condition index compared to oysters. It indicated stronger seasonal conversion of pelagic production into biomass and biodeposits. That is, they were efficient organic matter processing during bloom windows. So, coming to the condition index of European oyster, the condition index of European oyster at log ryan it was comparatively stable, but this is a snapshot, not throughout the year, and it is consistent. The condition index is consistent with constant immersion and more continuous food supply, so the condition index was quite good. So the health is quite good. Now, coming to the length frequency distribution of blue mussels in Zult. So these four panels they show the different habitats S1, S2, S4, and S5 respectively. And I would mention that S3 and S6 it didn't have any organisms, so they were excluded from this panel. So in case of mussels, we see that most of the mussels they were between the length of 35 to 55 millimeter. With adult, they dominated the population, but also we see recurring juveniles throughout the year. Uh, this supports the sustained filtration and biodeposition and ongoing shell formation. Now coming to the length frequency distribution of Pacific oysters at Zulu, again we see that most of the Pacific oysters they were between the length of 50 to 130 millimeter. So even at lower density, the larger individual they can store substantial carbonate per capita or per individual. So building a significant inorganic carbon reservoir. So for the European oyster length frequency distribution, we see that a typical subdural size spectrum, which is evidence of bed accretion and a persistent carbonate framework. These organisms they can help in stabilizing the sediment, and that also affects organic carbon preservation. So now this graph it is the sediment properties at Zult. So the top panel it shows the bulk density of sediment and the top 30 centimeter, and the bottom panel it shows the percent of mud that includes fine silt, very fine silt, and clay. So the chances of storing organic carbon increases when the percent of mud increases. Now, if we look into the top panel or the bulk density, we see that bulk density increases with depth a little bit. So it could be because of sediment compaction. And if we look at the grain size, if we look at panel C, at the bottom panel, panel C, this is S3. So S3 was taken as reference, which is intertidal bare sediment. There were no organisms present. We see that percent of mud was quite low, and if we compare it with panel A, B, or panel D, E, we see that the reefs, when there are organisms, they add roughness, so it increases small scale backfilling and also increases the residence time of the smaller particle. So it allows these smaller particles to persist beyond the surface layer. So coming to the this grain size and dry bulk density at log ryan, the left panel shows the dry bulk density of sediment. Again, similar to Zult, we see when the sediment depth increases, the dry density slightly increases, which is again due to sediment compaction in the deeper depth. And interestingly, the grain size, the mud percentage, it was quite high. It was between around 24-25%. So it indicates that a continuous submergence and slightly finer texture, it reduces physical shifting of the top layer or the surface sediment layer, and it also lengthens the particle and the sediment the seafloor contact, and it favors in organic carbon retention. So the subdermal bed, it showed more sustained coupling between pelagic supply and the benthic storage. Now coming to now we calculated how much organic and inorganic carbon it is stored within the biotic layer. So for that we calculated how much carbon is stored via their tissue formation, and how much organic carbon is stored via their tissue formation, and how much inorganic carbon is stored within their cells. So again, S3 and S6, they didn't have any biotic carbon presence on the top layer. So when we consider S2, it had high inorganic carbon levels because the Pacific oyster shells, they are quite dense, so they store a lot of inorganic carbon. And if we consider organic carbon, we see that S4, that is brown algae associated habitat, they had a lot of organic carbon because of the presence of brown algae. And we also saw that carbon storage, it varies with varying assemblies of organisms. Now, this panel is, I think, is quite difficult to understand because it has a lot of small graphs which is compiled together. But basically, let me give the summary of this panel. So it is the inorganic and organic carbon amount of inorganic and organic carbon present at Zult, and the orange line, this is the inorganic carbon or PIC, and the blue line, it shows the organic carbon or TOC. So in general, we see that mussel beds, the organic carbon, it peaks at the surface during spring. If you see the four seasons, there summer, autumn, spring, winter, and spring, and all six habitats, we see that during spring, the surface layer, it has increased amount of organic carbon, which readily declines with increasing depth, which indicates additive or oxygenated sediment, which is quite normal in the intertidal sediment, intertidal habitats. And if we consider PIC or inorganic carbon, it often increases with increasing sediment depth, as shell fragments, they are mixed and they persist as a subsurface carbonate memory. So where reef roughness is high, the amount of organic carbon penetrates slightly deeper. So when there is like a rough outlay of the reef, so the penetration of organic carbon below the surface layer, it increases. Now this is a heat map for the panel A. It shows the total organic carbon. Panel B, it shows the inorganic carbon, and panel C, it is the total carbon in these six habitats and at these different seasons. You can see. So what is interesting is we see that mussel beds, they remain inorganic carbon dominated throughout the year, but they show a strong organic carbon pulses during spring, which is because of increased feeding and biodeposition. Whereas the Pacific oyster reef, they show organic carbon richness during summer and inorganic carbon richness during spring. So the bare sands, it had a very less amount of carbon, organic or inorganic, and S6 or the subdermal reference site, it can match reef associated organic or inorganic carbon during the productive seasons, like during summer or during spring, but otherwise it is less in carbon. So this is due to because they are submerged, so there is longer residence time for the organic carbon to be stored at the surface layer. So one thing which is striking here is there are two peaks of organic and inorganic carbon, which is at different time. During summer, it is inorganic carbon, whereas during spring for oysters, during spring it was inorganic carbon rich, and during summer it was organic carbon rich. So we did a PCI analysis to understand all these variables and how these affect. And the x-axis, it shows basically that how much carbon is present overall. So the left side from zero it shows like how a higher amount of inorganic or organic carbon, and the y-axis it shows what kind of carbon. The up is carbonate dominated or inorganic carbon dominated and compact sediment, and the below from zero, the bottom down part, it is organic rich and looser sediments. So basically what we saw is across spring and summer, the sides they move left as the stocks of organic and inorganic carbon builds, and then they peel up when carbonate process dominates or peel down when organic trapping dominates. So reef changes between carbonate engineering and to organic carbon trapping rather than becoming a single static sink. So this is the inorganic and organic carbon at Loch Ryan. So this is a single time sampling, so a snapshot basically. So we see that at Loch Ryan, the total organic carbon it is elevated up to 15 centimeter before it starts declining, and the inorganic carbon it increases with increasing depth. So it is consistent with carbonate accumulation, and retention of organic carbon it requires time and micro barriers. So the cell hatch it creates roughness that slows near bed flow, and it promotes flocculation and boosts deposition efficiency. So with bioirrigation and microbial processing, some particles they are very quickly remineralized, while others they are sheltered and they are mixed and buried downward. So it favors the total organic carbon or organic carbon survival in steadier subtidal settings. So now coming to our second part, our second objective, which was to estimate the annual carbon budget of individual reef building organisms. First, we consider the cell growth. So this cell growth actually they generate carbon dioxide based on local alkalinity, based on partial pressure of CO2, and based on temperature. So at this current setting at North Sea, which is the total alkalinity varies between 2100 to 2400 micromole per liter, whereas temperature it varies between 0 to 30 degrees centigrade, and the partial pressure of CO2 it varies between 300 to 900 units. So here and actually they are generating carbon dioxide, they are not sequestering carbon. So based on the cell growth rate of different organisms, we calculated how much carbon they are producing via their cell growth. So we estimate that, and we considered the abundance of the organism as we observed in the field. So we took a mean abundance of blue mussels as 900 individual per square meter. For European oysters, we took as 80 individual per square meter, and for Pacific oysters, we considered them as 300 individual per square meter. So now when we consider this and we consider cell growth and we consider how much calcium carbonate they are gaining via this per millimeter or per centimeter of cell growth, we see that they can annually release a substantial amount of carbon dioxide. So if we consider blue mussels, because of their high density and because of their higher cell growth rate, which is about 35 millimeter average 35 millimeter per year in North Sea. So they can produce about 158 gram of carbon per square meter in a year. Whereas Pacific oysters, though their density is less, but they have very high cell biomass. So therefore, they are like their individual production of carbon dioxide is quite high. And if we consider European oyster, they produce least amount of carbon dioxide due to slower cell growth rate, which is between 15 to 20, 17 millimeter per year, and because of their decreased abundance compared to blue mussels or Pacific oyster. But we should remember that carbonate, they builds the habitat, and the habitat, they modulates the organic carbon retention. Now coming to community metabolism, we did it using benthic chambers or incubation chambers. Both light and dark conditions were measured, and we did the experiment with in situ sediment samples, and it was done within a mesocosms, but it doesn't capture the annual community metabolism. It is a single time experiment, so that was a limitation in our experiment. So I'm not going into details about the methodology, but you can ask me later. So basically, we converted the oxygen dynamics, or how much dissolved oxygen they consumed during daytime and during night time, and then we converted it to carbon mineralization rate, and we estimated how much carbon they are generating per square meter per year. So in this graph, you can see that basically they are generating carbon throughout um, like respiration. It generates carbon continuously, or it is a CO2 efflux. It is not fully balanced by daytime photosynthesis. You can see like their net um, carbon dioxide producers, even if you subtract the photosynthesis or production of oxygen during daytime, still they are producing carbon dioxide. So, um, in case of muscle bed, the dense muscle bed, they show higher per area respirations because obviously their density was high. And in case of Pacific oysters, they show strong efflux of carbon dioxide because their size was larger, so they generate more carbon dioxide. And in case of European oyster, they exhibited a steadier production of carbon dioxide, which was lower per area efflux under subtidal condition when we compare it to blue mussels or Pacific oysters. Now we wanted to account for the organic carbon production by biodecomposition. So we did a experiment with mesocosms where we used flow-through system, and we also mimic the settings of subtidal and intertidal uh, habitats. And here we used all three organisms: blue mussel, Pacific oysters, and European oysters. And we wanted to isolate the pathway where filtration leads to fishy or pseudofishy production, which then accumulates on the seabed. And here we used the realistic temperature and realistic food availability. We didn't add any food. So, and the realistic flow rate of natural seawater. So we acclimatized the organism for 3 days before we started the experiment, and then we conducted the experiment for 21 days. And you can see the mesocosms where we did the experiment, and this is how the biodecomposition it looks basically. So coming to the results, you can see the top panel A, B, C. It shows the intertidal mesocosms, intertidal habitats mimicking mesocosms, and the bottom panel it shows the subtidal habitat mimicking mesocosms. And we also use three different temperature range to understand if increase in temperature can increase their feeding also increase the biodegradation. So therefore we used three different temperature range, that is 12 degree, 17 degree and 22 degree centigrade. And then we estimated how much carbon is present in the biodeposits, and we calculated the annual carbon production via biodeposition. So here we see that biomass density is the first order driver. That means that more filter feeder there is, so there is more package, more carbon into fast sinking biodeposits. So for mussels, let's say mussels, they had higher abundance, so you see they can store more than 1 kilogram of organic carbon per square meter of area. And then we wanted to understand, we did a 2 and over to understand the influence of species, tide and temperature on biodiversity. So here we can see that the species and tide, they had significant influence on the biodiversity rate. And in case of subtidal, in subtidal conditions, they enhance the flask across species, but temperature, at least within our temperature range, it didn't influence the biodiversity that much. But I would say that this experiment was carried out in October, so the amount of food which was available to them, to the organisms, it was quite low. It was about um, it was quite low, less than 2 micromole of chlorophyll per liter. So yeah, so this was one drawback. So sorry. So basically, we see that exposure, it matters. The subtidal conditions, they sustain feeding and they reduce wave-driven export of crop layer, and they increase the share of biodeposits that actually contact and remain within the sediment. So within, so yeah, so then we wanted to understand what is the carbon remuneration rate at different depths of the sediment. So we did a simple assay which is called aroma or rapid organic matter assay. So here we constructed these small cages within our workshop. So we added known amount of agar within these cages, and we buried these cages at different depth of the sediment. And we wanted to understand how much carbon is getting mineralized at different depths. So we analyze it between 0 to 25 centimeter of sediment depth. So here you can see that the control which is bare sediment or unvegetated sediment, and the reef adjacent or reef associated sediment. So the open sand or the control, it sustains relatively uniform activity. You can see that the carbon degradation, it doesn't change much with increasing depth. So it remains almost more or less same. Whereas in case of reef adjacent sediment, the top layer, the carbon degradation is quite high, but with increasing depth, the carbon degradation is quite less. So it is consistent with reduced permeability of sediment and more refractory material which is being buried below the first cycling zone, that is top 5 to 10 centimeter. So sorry. So coming to the summary. So in case of intertidal shellfish reef, we saw that mussel beds, they are organic accelerators, and they maintain their carbonate structure, and they convert the bloom organic carbon to biodeposits while sustaining a shell bank. In case of Pacific oysters, they are organic carbon rich in calmer, warmer summertime, and they are inorganic carbon rich in spring. And in case of macroalgae, they add short-term organic enrichment, but they can be easily reset by storm events or by seasonal changes. And in intertidal setup, the main constant remains sediment permeability and short resident time of organic carbon. So in case of lock rand, this is subtidal sediment or subtidal habitat. We saw that subdural oyster bed, they show how continuous coverage and slightly finer sediments, they lengthen the residence time, they create frequent small deposits and maintain a stable carbonate framework, which helps to trap the organic carbon. And the stocks are in kilogram scale. They are not in tens of kilogram like seagrass or salt marshes, but the probability of organic carbon survival in the subtidal regime it is quite high and incremental policy relevant gain. So for take home message, we see that shellfish reefs they are process engineers. Carbonate builds the scaffolding, then filtration and biodigestion processes. They load the bed with organic carbon, whereas hydrodynamics and the texture of sediment they decide the retention of this carbon. So for accounting, I would suggest to report them in both organic and inorganic carbon and separately report them. And for practice, we should consider the organism density and the placement of organism very carefully. So, for example, higher biomass and subtidal setting, it enhances the delivery of organic carbon from water column to the seabed, and also their retention time is increased. In case of mixed mussel and oyster beds, combine strong delivery with durable structure of the reef, and the climate dividend is more or less nuanced, but it is measurable. So modest in magnitude, and it is basically tied to biodiversity and habitat function as well. So I will stop here, and if you have any question, please ask. And especially if you have any questions regarding methodology or any data related uh, results related questions, you can ask or you can write to us using this email ID. And I would like to thank Bill for his cooperation from Edward University, and I would also like to thank our funding agency, EFN. So